Hello everyone. Chapter 3 is going to be an overview of the cellular structures of bacteria, and we'll touch a little bit on the archaea as well. But this figure here in your textbook is a really good summary of all of the structures that we're going to be covering in this lecture. Morphology and arrangement are going to be a really important way that we characterize bacteria. So let's talk about what these mean. So when we say the word morphology, we mean the shape of each individual bacterial cell that we're looking at under the microscope. Not to be confused with arrangement, which is how when you have multiple cells together of the same type, how they pattern themselves as a group is going to be their arrangement. And so all bacterial smears, when we look at them under the microscope, are going to be characterized by their morphology and their arrangement. So the arrangement comes from two different questions. How was the cell divided? Right? When we talk about the planes, think about the planes of the body that you learned in A and P. Right? If you divide the body down the center and you have lateral portions, you divided it down the sagittal plane. We have the coronal or frontal plane if you end up with anterior posterior divisions. So the shape of the cell, how it's divided, and whether those daughter cells remain attached or whether they separate are going to be the characteristics that determine the morphology and arrangement. In bacteria, we have these basic morphologies. So when we say a coccyx, or cocci would be the plural for that word, is a sphere. Coccobacillus would be sort of an elongated sphere, not quite as long as a rod, which is the word bacillus. A vibrio is going to be sometimes referred to as a bent rod or a comma. We have two kinds of spiral morphology: spirillum, which is really usually a little bit fatter in circumference, and it's a little bit more rigid than the spirochetes, which are going to be very thin, but also very flexible. So when you see these guys under microscopy, you're going to see spirochetes are sometimes going to look like they're bending as they move through the medium, and because they're flexible. And then these pleomorphs, these are going to be those that every time they go through cell division, they actually change their shape. So these are really tough when it comes to diagnosing because they're very difficult to just determine morphology, which is one of the first things we do when we look at them under the microscope. The pleomorphs are going to be including things like the mycoplasma. So if you recall, we introduced mycoplasma bacteria, those that don't have a cell wall. So since the cell wall is going to be an important part of what determines their shape, not having a cell wall is partly how you get this pleomorphic groupings. Your book has a good table that shows a little bit of the variation within each of those groups. So table 3.1. So we use the general morphology terms, but realize there's some variation. Arrangements, recall again how you divide the cell will determine whether they, which direction the pattern is going to go, but also if they stay attached or not. So if you have a coccus bacteria that once it replicates, it lets go of its sister or the other daughter cell, then we would use the same word for the arrangement as we would for the morphology. Meaning if it's a single sphere in a mature smear, we would use a morphology of coccus and arrangement of coccus. If they retain at least a pairing, we would call that diplococcus. If they tend to make long chains, we would call that streptococcus. If they form what we call a tetrad or a sarcina, it's because they just used two perpendicular planes when they divided. And then, of course, we have the famous staphylococci, which is basically we have these random planes. So you can take a sphere and divide it down the middle in any which direction, and you're still going to get the same shape of daughter cells. And so when you get these big clusters, in fact, the word staphylococci means grape-like cluster. There's also another group called the micrococci, which we won't see very often, but the staphylococci is going to be very commonly seen. So let's look at some examples of each of these. So again, arrangement cocci, you can see here the majority of the cells are actually separated from their neighbors. So keep in mind, anytime you look at a smear, number one, they're under the microscope here, at about 1,000 X, which is the highest we can get on our compound light microscopes in the lab, you're going to have to use a little bit of a judgment call to decide which arrangement you're looking at. So always look at sort of the general pattern. What's the majority of the cells that you're looking at? What are they doing? So here the morphology would be Coccus because of the shape, and the arrangement, the pattern that the group is doing, would also be Coccus or cocci. And don't worry too much about Coccus being singular, cocci being plural. As long as you spell it correctly, you can use either of those terminologies. Diplococci. So if you look closely, these kind of look like little figure eights, and that would be paired spheres for the arrangement. Streptococci would be chains of spheres. You can see down here on the left bottom corner, that electron microscope diagram, those almost don't even look like spheres, and that's because you're looking at a very young culture. They haven't quite reached their mature shape just yet. So it's really important when you look at bacterial smears that they have been prepared at the right time and that those cultures are mature. Tetrads are going to be little 4-packs. These are rare. You're not going to see these very often, as well as the Sarcina, which are basically 8-packs. Bacilli, of course, if you recall, the word bacillus means rod. So if you have a rod-shaped bacteria, the only way that you will get division across this cell is across what we call the transverse or horizontal plane, right? Across the short, going across the long axis. And so when these cells divide, they would go away from that division, right? You would never divide this cell down the long axis and have two sort of stacked cells next to each other. So when you get arrangements of bacillus, again, if they tend to let go and they exist as singles, the morphology is bacillus and the arrangement is bacillus. If they pair up, we would use the word diplobacillus. If they make long chains, we would say streptobacillus. And if they do something somewhat unique, what we call V-shape would be sort of the first stage of this type of arrangement, where basically they, instead of staying in the same plane with each other, they would basically hinge. And so you can see when those two cells, when that cell divided and it had a sister cell, it just hinged. And then when those two divided, they hinged again. And so what they end up doing is sort of accordion across each other, and they end up with these little stacks, what we call palisades. So V-shape is usually the very immature version. Palisade is the mature version of that type of arrangement. But again, those are usually very rare. So some more examples, bacillus, and this is a very heavy culture, which means you're going to see so many cells, sometimes it's hard to distinguish which arrangement you're looking at. So sometimes the electron microscope micrograph is easier to interpret. So the red cells down here in the bottom left corner are pretty clearly singular cells, right? They're just free-floating single cells. The larger micrograph that you're seeing, these are very large bacillus cells, and there's so many, it's almost hard to tell. So it's, again, somewhat of a judgment call when you start making morphology and arrangement determination. Diplobacillus, again, paired. Now, you do see a couple of cells in there that are just singles, but the majority of them are pairing up. So always go with the majority. Streptobacillus would be those that, again, are going to be chains of rods. What's happened so very oftentimes in streptobacillus arrangements are that these rods, which really, if they were individual cells, would be sort of rounded off at the end, they almost tend to sort of square off with each other. And so they end up looking like long strings of spaghetti, so to speak, when you're looking at them under the microscope. So don't let that confuse you if it's hard to see those individual cells in those long strings. This would be streptobacillus. Now, there is no such thing as Staphylococcus because you would have to have random planes of division. So keep that in mind. Arrangement V-shape and palisade. Again, if you look in here, you'll see a few that are in that V-shape. Here's an example here. So V-shapes here, but everywhere else you see what starts to look like little stacks, right? And those stacks are going to be that palisade arrangement. Here's a good stack of 3 right here. So again, they didn't get stacked that way because they divided down the long direction. It's because they hinged and then they just sort of kept accordioning back and forth with each other. Pleomorphs, again, the arrangement's always just going to be the same word that we use for the morphology. Pleomorphs do not stay attached to their with other daughter cells. So the arrangement and the morphology would be the same term. The spiral morphologies, we mentioned the spirillum are usually very rigid, and they're usually a little bit fatter in diameter than the spirochetes. Of course, you're rarely going to see them together on a slide where you're comparing the thickness of the cell. But you can see the spirochetes have a little bit tighter of a curl. They're also slightly flexible. You can see that they, like this cell here, is sort of bending as it's moving through the medium. And so spiral morphology, there are some spirillum that can link in a chain. They'll have 2 or 3 linked together. But for the most part, spiral morphologies are always going to be the same word for morphology as arrangement. And you would just use these words. So spirillum for morphology, spirillum for arrangement, because they're singular cells. The same thing goes for the Coccobacillus. These are sort of elongated spheres, something between a coccus and a bacillus. These are fairly rare. You won't see these very often, but these also tend to always be singular. You usually won't see these in chains or rods, pairs or chains. So they would use the same word for arrangement. Vibrio, same thing. So they vary. Here we're probably just catching them in the act of binary fission. They just haven't let go yet. But you can see these are bent rods. And so this would be, for example, Vibrio cholerae, the organism that causes cholera, is a Vibrio morphology. Pleomorphic, again, we said is going to have a variety of shapes. And so you can see here, what looks like maybe a Coccobacillus, some bacillus. Here's, it looks like a golf club. It doesn't look like any of the others. But some of these can be really unique. They can look like stars. They can have pretty much any random shape. And so pleomorphs are really problematic for diagnostic purposes. Now, if we move on to the cellular structures, you're going to see that cells are protected by some surface layers. And so if you put all the layers that are on the cell together, we would call it the cell envelope. But that envelope is usually composed of 2 or 3 distinct layers, one of which would be the cytoplasmic membrane. Of course, we usually call that the cell membrane. It'll be just like the one that we learned about in eukaryotic cells, which is made of a phospholipid bilayer. Superficial to that will be the cell wall. So we talked about cell walls being an important structure. We're going to learn what those guys are, how those are built today. So recall we mentioned peptidoglycan was the specific structure or substance that bacterial cell walls are made of most of the time. So we're going to talk a lot about the cell wall. And then the outer membrane is a special layer that's only found on gram-negative bacteria. So we're going to look at what that looks like as well. So if you compare the 2 main groups of bacteria, we're going to be looking at the gram-positives versus gram-negatives. What determines their gram status is actually the structure of the cell wall. So if you look here on the left, we're going to see gram-positive cells have a very thick layer of peptidoglycan. The ones on the right with the gram-negatives have a very thin layer of peptidoglycan and that outer membrane. So we're going to look at the structures associated with each of these. The peptidoglycan layer itself, if you recall, peptido meaning peptide. So this if you want to think of the cell wall kind of like a brick wall, you have bricks and then you have mortar that attaches the bricks to each other. So the bricks here are what we call the NAG and NAM chains. These are the sugar molecules. These are 2 different kinds of carbohydrates that are then linked together with proteins. And the proteins would be these tetrapeptide cross bridges. And so when they say tetra, that just means 4. There are 4 amino acids that serve as the link between each of these rows of carbohydrate NAG and NAM chains. And so when you look at the cell wall, you can see there's gaps, right? There's spaces between these. So I like to think of this sort of more like a scaffolding. It's not a solid brick wall with no gaps. This is going to be immediately superficial to the cell membrane. And so these gaps that you see are big enough to allow nutrients and water and other substances through, but not big enough for the phospholipid heads to move through. So the purpose of a cell wall is to protect the membrane underneath from osmosis. Because if you recall, phospholipid membranes are susceptible to osmosis. If you have a cell wall, you're cell membrane won't burst when you get exposed to a hypotonic environment. So that's really the purpose of the cell wall for the organism that has it. And so keep in mind, we keep harping on the cell wall because this is a major target of antibiotics. So just knowing how it's made, what it's made of, is going to be an important factor. How that cell wall looks in the two different Gram groups would be, again, Gram-positive bacteria have a very thick layer of peptidoglycan, and it's so thick that it actually needs more than just the peptidoglycan. It actually needs sort of, I like to think of these as like rebar, right? Bars of reinforcement. So teichoic acids are these substances that sort of just anchor the bricks themselves, and then lipoteichoic acids are those that anchor that thick layer all the way down to the cell membrane underneath. Gram-negative cells have that very thin layer of peptidoglycan, and so they actually have a third layer, what we call the outer membrane. And that outer membrane is made of half phospholipid and half lipopolysaccharide. So the LPS layer is the part of the cell that actually is very toxic to the human body. And so we actually call this endotoxin. You're going to hear a lot about endotoxin or LPS. Sometimes they'll refer to it as lipid A. All of those are referring to this outer layer, what they call the outer membrane of gram-negative cells. This is the thing, this is the reason why, in fact, that the very first thing we do is determine the gram status of a bacterial infection. If you have a patient that has a raging gram-negative bacterial infection and you give that patient antibiotics, what's going to happen is you're going to kill the bacteria. It's going to release the lipopolysaccharide into the patient's body, and it's going to flood the body with something that triggers really high fever and shock and other things that we'll talk about when we get to the immune system chapter. It's basically going to trigger the immune system in a very negative way. And so keep that in mind. This is the reason why we do a gram stain to start with, is this substance right here, LPS. So keep that in mind. That's actually part of that outer membrane, which is on the surface of gram-negative bacteria. So how do we do the gram stain? Well, based on the structural differences between the two groups that we just talked about, the gram stain will help us differentiate between those two groups. And so the first thing that's done on an unknown bacterial smear would be to apply what we call crystal violet, which is going to be the primary stain. Now this is just a basic stain. It's going to stick to anything that's acidic. And if you recall that lipoteichoic and teichoic acids of the gram-positive cells, they have a very heavy layer of those acids. And so they're going to take up that dye quite readily. Actually, both gram-positive and gram-negative cells will take up this dye. The fatty acid tails of the phospholipids of the cell membrane will also take up this dye. And so all cells will stain purple, basically, after the first step of the gram stain. The second step would be what they call the mordant. And you think of mordant like mortar, it's going to be like glue. And the Gram's iodine is used in this step. So the Gram's iodine doesn't stick to the cell, it sticks to the crystal violet. And so whatever crystal violet has attached itself to the cells in the first step now gets adhered to by the iodine. And so then you end up with what's called the CVI complex, or the crystal violet iodine complex. Now, the crystal violet was small enough that it could be rinsed out potentially of the cell, but the Gram's iodine creates basically a glue, and it keeps that crystal violet and sort of helps it adhere to the structures where the crystal violet is. And now it's a larger complex that can't readily be removed from that outer membrane of the gram-negative cells. The third step would then be the decolorizer. And this is an alcohol step. This is that accidental step that happened with when Christian Gram discovered this process. He accidentally dropped alcohol on some of his slides. So the outer membrane is actually very susceptible to alcohol. And so the gram-negative cells will then be decolorized. And this is the differential step. If you're a gram-negative cell, that alcohol will punch holes essentially in the outer membrane. And in between each of these steps is actually a water rinse. And so when you rinse these cells with water, any dye that hasn't attached to something, it gets rinsed away. After this decolorizer step, the holes in the outer membrane then will allow that crystal violet to get removed from the cell. The final step then, because we've just decolorized the gram-negative cells, if there were any gram-negative cells on that slide, they would be invisible at this point. So we've got to have a counter stain, and the counter stain is a red dye called safranin. Now, safranin is also a basic dye. It's going to stick to everything that the crystal violet stuck to, including gram-positive cells. So both cells will take up the safranin. It's just that you won't be able to see the pink or the red on top of the purple. So if the gram-positive cells are there, they're gonna stain purple. They're not going to look any different. But if the gram-negative cells are there, we decolorized them in the previous step. Now they're going to look pink. So anything in the purple family is going to be gram-positive. Anything in the sort of red to pink family is going to be considered gram-negative. Keep in mind, there are some of those bacteria we've talked about. There's one group, the Mycoplasma, that naturally don't have a cell wall. So this creates a problem when we're doing gram stain. They also have sterols. So if you recall, in our cells, we have cholesterol, which helps our cells not melt in the heat or get too fragile in the cold. Most bacteria don't have sterols. And so we're going to talk about some of the antibiotics that actually target sterols. Well, Mycoplasma do have sterols, and that's one way that they can stabilize their cell membrane because they don't have a cell wall. There's one group of pathogens we'll talk about that belongs in this group, walking pneumonia. One of the reasons why it's hard to diagnose walking pneumonia is because Mycoplasma are not going to stain properly in the gram stain process, and that's because they lack a cell wall, but they're also pleomorphic, as we just learned, so they're going to be difficult to diagnose no matter what. There are actually also some bacteria that have a cell wall, but then they lose it during part of their life cycle. And so this becomes what they call the L form. And this is really problematic for infection. So picture you have a patient that has a persistent infection. If they take a sample that they try to do a gram stain, but it's during the L form stage where the cell no longer has a cell wall, clearly the gram stain is going to be give false data. And so this is a big problem for diagnosing those types of infections. And it's also a problem for antibiotic resistance because, as we've talked about multiple times, the cell wall is going to be one of the major targets for antibiotics. So if they don't even have a cell wall when you're being treated with that antibiotic, it's not going to have any effect. Some groups, like those that cause C. diff infections and anthrax, can form what's called an S layer. And this S layer is sometimes referred to as cell armor or chainmail, right? It's just an extra protective layer. And this usually only gets formed on the cell when they're exposed to very hostile environments, right? And very harsh environmental conditions, hot, dry, nutrient free kind of environments. But where you would find this would be sandwiched between the cell wall and the structure called the glycocalyx, which we'll talk about next. So glycocalyx or glycocalyces, plural, are, if you break down that word, glyco meaning sugars. So this is really just sort of a coating of polysaccharide that prevents the cell from drying out. It helps them sort of hide from our immune system. So when we say it's camouflage, we mean from phagocytes, right? If you think about it, these guys are basically coated in sugar. That polysaccharide is something that our immune system would typically just think is not harmful, and they would ignore it and they would not destroy them. So this camouflage is going to be an important virulence factor that we talk about. It's also, because it's sugar, it's sticky, and it can help provide some adhesion. That's not just for them to be able to make biofilms, but this is also a problem for those species that can grow on medical devices like intubate catheters and pacemakers and things like that. But usually glycocalyx comes in one of two forms, what we call a capsule, which is going to be a very thick, very organized, regular kind of repeating pattern that surrounds the cell, or it's going to be a slime layer, which is very water-soluble, very irregular. This is going to be an important thing that we see in some of the fecal bacterial infections. Now, flagella, we are familiar with flagella on a sperm cell that we've studied in A&P, but a flagella in a bacteria looks very similar, but actually is built quite differently. So the filament is actually the tail portion. The hook is the rotating attachment point there for the filament, and it attaches it to what they call the basal body. And the basal body is a series of protein rings that attaches the hook of the flagellum into the cell membrane and the cell wall, or outer membrane if it's a gram-negative cell. So there's basically the three parts of the bacterial flagella, and that hook allows the flagella to basically rotate like a boat motor. They arrange their flagella differently, and this is actually something we can use to diagnose different bacteria. And so if they have a single flagella, if you're a bacillus shape, you would have it at the end. You would never see it coming off just at the center of the long axis there. It would always come off at the end, and they would call that monotrichous. So trich is the root word that means hair. Lophotrichous means tuft, and so you can see there's multiple flagella all coming off at one end of that cell. And when they went to use these flagella, they would basically lay them all together, and they would act like one big flagella. Amphitrichous, so that root word amphi means both, right? Think amphibians live both in the water and on the land at some point in their life. And so what we call polar singles, just what it sounds like. At each end, you're going to have a single flagella. Now, this is a very common arrangement for the spiral bacteria, but you may also see this on Bacillus. Peritrichous, peri means around, so here you're going to have dozens of flagella all the way around the cell. And these also would sort of all lay in the same direction and act like one big flagella when it was moving around. So bacteria and eukaryotes, they might both have flagella, but they're actually built very differently. So again, you have the filament, the hook, and the basal body on the bacterial flagella, but you can also see it's actually an external structure. It's attached at that cell membrane, and then it sticks out away from the cell. In eukaryotes, it's actually part of the cytoskeleton. It's actually made of those hollow microtubules that are also found at the core of a cilia, and they sort of get pulled back and forth, and they undulate. So bacterial flagella rotate, eukaryotic flagella undulate, but also look where they are. In eukaryotes, they're actually an internal structure. They may project away from the cell body, but they're actually within the cell membrane. So they're actually internal structures. What they're made of is also different. So the little proteins that make up that filament are called flagellin, and the hollow tubes that make up the eukaryotic flagella are made of a protein called tubulin. So there's a little compare-contrast to looking at how those two differ from each other. If you were to see them on the cell, the eukaryotic flagella, you would only ever see the single flagella arrangement, the monotrichous. If you see multiple hair-like structures that are short, they're probably cilia. Now, don't confuse this with a peritrichous bacteria. If you were to see a cell that is coated in flagella, it would be a bacteria. And think about the size scale here. This is a eukaryotic cell. Picture that cilia covered with millions of bacteria. That's how small the bacteria would be. So you would never see cilia on the surface of bacteria. Okay, cilia are exclusively found in eukaryotes. Bacteria do have another kind of projection, what we call a pilus or a pili for plural. Sometimes they'll have more than one. But this is basically like a hollow tube. I always like to compare it to the little chute that you use when you go through the drive-through at the bank, where they send the little box back and forth where you can exchange things, which is exactly what these bacteria are doing. They're exchanging DNA in a process called conjugation. And so these are not motile. These are not tails. These are not flagella. They're not moving around using these. They are just using them as a hollow tube for the process of conjugation. And what they're sharing through that tube is a plasmid, which is just a small, round, circular stretch of DNA, which essentially, of course, represents a gene. And the gene allows that bacteria to then make a protein it couldn't make before. So sometimes those plasmids are transmitting information to make maybe an enzyme that allows them to use a food source they couldn't before. Those plasmids very oftentimes are going to be called R plasmids for resistance, because many times the genes that they're sharing is the necessary information for that bacteria to make perhaps a protein pump that they can then use to expel the antibiotic from the cell before it can kill them. And so they call them R plasmids for resistance plasmids. And we'll talk a lot more about that this semester. Fimbriae are sometimes confused with cilia because they're very short projections coming off of a bacterial cell. But keep in mind, these are going to be different than cilia. These are non-motile in the sense that they're not moving the cell around. They are sometimes described as biological Velcro. So they're going to be very sharp, sort of bristle-like, if you think, little hooks on the end that allow them to stick to each other and to surfaces. And this is another way that they can make biofilms. So a capsule can make them sticky and allow them to stick, or biofilms can be formed from fimbriae. And so we say mostly non-motile here because fimbriae have been known in some species. So if they're laying in a biofilm together, they have been found to sort of throw that fimbriae out like a grappling hook attached to the neighboring cell and then pull that neighboring cell close enough together that then they could then form a pilus and start sharing DNA. And so they're basically a little projection that allows them to stick to their neighbor. Once they're in that biofilm, we've mentioned many times that bacterial diseases, most of the time, 99% of the time, requires a biofilm. And that's because bacteria sometimes behave differently once they have communicated with their neighbor. And so this is not just a problem for human infections, but the fact that medical devices, the surfaces, if they're not properly sterilized, can become coated and contaminated with bacterial biofilms, which then becomes the source of the bacteria. And so we just mentioned when they're conjugating, they're going to share information. And so very oftentimes what happens once they reach a threshold, when there's enough bacteria in that pile, so to speak, they start communicating with chemicals. And so this little chit-chat they call quorum sensing. And that actually triggers them sometimes to make toxins they couldn't make before. Very oftentimes bacterial diseases, not only do they make biofilms, but they cause their damage through toxins. And those toxins are very oftentimes proteins. So they just needed conjugation to get the secret recipe. Now they can make that protein, and that protein is a toxin. Sometimes, again, we mentioned the DNA that they're sharing is an enzyme or a pump or something that allows them to resist or survive being exposed to that antibiotic. So we'll talk a lot more about quorum sensing. There's a lot of research into developing drugs called quorum quenching drugs. Because we have so much antibiotic resistance out there, we've got to find new ways to go after them. Ribosomes, if you recall from your prereq, these are where we do protein synthesis. And so ribosomes are going to be slightly different between prokaryotes and eukaryotes. Prokaryotes are going to have what we call an 70S, and this is basically a measure of sedimentation. So the S stands for a Svedberg. That's the unit for if you put a sample in a centrifuge and you spun it down, how quickly did it settle to the bottom of the tube? That's called the sedimentation rate. And so prokaryotic ribosomes are what we call 70S. They're actually made of two separate little subunits. Now, this math doesn't add up. It's not supposed to, right? So when we sediment something, it's based on the shape and size of the surface area of that structure. And so if you were to break up these two subunits, there's a large and a smaller subunit where the ribosome comes together. If you separated those out, it would be a 50 and a 30 S subunits. If they enzymatically broke them down and spun them out, they would come out at two different sizes. And so these become the targets for antibiotics, those little subunits, because they are little proteins and they have a different target. Now, eukaryotes also have to make proteins. All living things do. But the eukaryotic ribosomes are actually a different style. They're a little bit bigger. Most of them are going to be 80S. And so again, if you broke those into two smaller pieces, they're not going to add up. They're going to be 60 and 40. But that gives us something that is slightly different from the bacteria. So this again, antibiotics, some of them work on cell walls and some of them work on ribosomes. Because they're slightly different, it makes a good target. However, this most word is the key thing here. Not all ribosomes in a eukaryotic cell are 80S. Some of them are 70S, which is just like the prokaryotic ones. And so in the mitochondria, if you recall, that's the powerhouse of the cell where we make ATP. There's a lot of protein synthesis going on in a mitochondria. And so they have their own independent ribosomes. Turns out they're just like the prokaryotic ones. And so this becomes a problem because some antibiotics, if they target those 70S, then the mitochondria also become damaged with that antibiotic. And so usually this is where side effects in some antibiotics come from. So if you target the mitochondria in your cells while you're taking antibiotics, then you're going to make less ATP and potentially have fatigue. And of course, this is usually associated with long-term antibiotic use. But again, that makes this not a perfect target. We want a magic bullet. We want something that hurts them and doesn't harm us. And this becomes a problem. Now, endospores are also a really big problem in medicine. And these are basically a structure that bacteria can use to defend themselves against hostile environments or unfavorable conditions, such as extreme heat, complete lack of moisture. So, living things need moisture to metabolize and grow. And when you take that moisture away, they have to either die or protect themselves. Very low temperatures, freezing, radiation, chemicals, and even just lack of nutrition can cause cells to go into this defensive mode. And so what they do is they basically protect a copy of their DNA in this structure called an endospore. And so this area here would be the endospore, which is basically just a little compact area of nothing but DNA. And the rest of the cell would be called the sporangium. And so in some species, they've actually found that once the endospore has formed, the bacteria actually produce little toxins, crystals. This species, for example, creates crystals that contain a toxin called Bt toxin. This is just an abbreviation of the scientific name, Bacillus thuringiensis. But this Bt toxin has actually been, it's an insecticide that's been used for decades by organic farmers. We'll talk about that a little bit more when we get to the environmental chapter. But today we're going to focus on spores. And so producing an endospore, you have to go through a set of steps called sporulation. And this takes about 8 to 10 hours for the cell to form the endospore. But basically, this is not reproduction, right? Even though it starts out with copying the DNA. You need 2 copies of the DNA, one of which is going to get protected in the endospore, and the other of which is going to just disintegrate. So basically, you copy the DNA, and then you surround it by this thing called the spore coat. The spore coat is a really hardy layer of calcium and a substance called dipiclinic acid, which basically produces this spore coat. And within that spore, the DNA basically gets dehydrated. So it puts it in this sort of a state of rest, if you will. And then the rest of the cell just sort of disintegrates. So again, we keep mentioning this is not reproductive, even though it copies the DNA. And why it's not reproductive is because you don't end up with more cells than you had originally. What happens is an actively growing cell for bacteria is usually referred to as a vegetative cell. I know that sounds counterintuitive by how we use that word when we describe patients. But a vegetative cell is an actively growing, living, metabolizing bacteria. It can form one endospore, and when conditions get better, that one endospore can germinate or come back to life and sprout, so to speak. That's why they use the word germinate, back into one vegetative bacterial cell. So we didn't reproduce the cell. We just kind of hid a copy of itself away and then let it come back to life when conditions got better. So that germination stage is really fast. It can take, just an hour and a half to come back to a fully formed, fully functioning vegetative cell. And again, we can diagnose which bacteria we're looking at in a patient based on the position and location of those endospores. So if it's just smack dab in the middle of the cell, it's going to be called central. If it's toward the end, it's going to be called subterminal. And if it's at the very end of the cell, it's going to be terminal. So those are some diagnostic characteristics. So why we care so much about bacterial endospores, because these are so hardy. When we talk about them being hardy, we're talking about thousands of years potentially. I think your book mentions that they now have evidence of a Bacillus endospore that was viable from a salt crystal that was aged at like 250 million years. Prior to that data, the longest lived data we had was an endospore that someone had grown from a sample that had been taken from a sarcophagus with a mummy that had been thousands of years old. Of course, a lot of critics said no, it just came in with the dirt as they were extracting the artifact. The only really confirmed long-term study with an endospore was a gentleman who, at the beginning of his career, put some endospores away, and right before he retired 34 years later, grew one of those endospores back in the lab. And so we only had confirmation that 34 years was the longest. You know, he retired, he maybe passed on some of those samples to other researchers that have probably extended that, confirming that data. But why we care about this is because endospores, because they're found in the soil, pretty much all the bacteria that can produce endospores, if we can if we can sterilize and kill endospores, then by default we will kill anything that's weaker than an endospore. And so there are literally 5 diseases that we are concerned with, not just in medicine, but in food canning practices that protect us from endospores. So for example, anthrax, right? So when we talk about people opening up their mail and being worried about finding the powder, that powder is actually, you know, if it's been developed for a biological weapon, it's basically full of endospores. And if you breathe it in, that endospore will get into your respiratory tract, it will germinate in your body, and it will grow and cause anthrax. Botulism, right? This is why we have proper food canning standards, so that we can get rid of any of the bacteria that came in as endospores on the dirt that entered, you know, for example, canned vegetables, right? They came out of the garden's full of dirt. If the dirt was not removed properly from those vegetables, when you can them, you basically be putting that bacteria in an environment where it would flourish, right? So there's a reason why they tell you not to buy canned goods that are swollen or dented. What you're seeing there is if it's potentially not canned properly, then that bacteria is growing inside of that anaerobic can and producing gases, and that's what's warping the can, right? So now canning is much more likely to get botulism if you're home canning and you don't follow instructions. But botulism, again, endospore disease. Tetanus, endospore disease, right? The old wives' tale that you get tetanus from stepping on a rusty nail. Actually, it's not the rusty nail at all. The nail is the culprit because it can cause a deep wound. It's rusty, which means it just has been out exposed to the elements long enough to have a roughened surface where the oxidizing has occurred, and that roughened surface probably contains dirt. And so if you step on a rusty nail, it's not the nail that did it to you, it's the bacteria that came in as an endospore on that rusty nail. And then can get into the deep anaerobic wound and grow and cause the disease tetanus, which we'll talk about both botulism and tetanus when we get to the nervous system diseases. Gangrene, right? Again, wound with dirt creates a situation where you could have a problem, and it's because of endospores. If endospores get into the deep anaerobic wound, then it can grow and cause a catastrophic disease, which is gangrene. C. diff, this is becoming a bigger problem today in medicine, but it's the endospores that are very hardy. They get transmitted through the air, through the bedding and on surfaces throughout the hospital, and it's becoming a bigger problem. But this is an endospore disease. So again, we worry about endospores because they're the toughest thing, with the exception of prions, which we'll talk about later in this semester. But if you can kill an endospore, you can pretty much kill anything, and that's why we aim for the top of that list in endospores. And here you're looking at it. These 5 diseases are generally why we sterilize everything. We autoclave everything in medicine. So in summary, we've talked about the domains of life back in chapter 1. Again, I've kind of highlighted bacteria and eukarya here more so than archaea. They're no pathogens, so we're not really too worried about that group. But if you pay attention, there's some purple font here that is an addition to what is in this text. This is a table from your text, but it has a little bit of an addition there. And then finally, we've talked about, your book also talks about there's 4 different classifications or major divisions of prokaryotes just based on their cell wall structures. This diagram, what you're looking at, is a slightly different arrangement. This is actually dividing up all bacteria into groups based on their RNA sequences. So by no means are you supposed to memorize this or that other 4 divisions of big complicated names of the different groups of bacteria. First, let's look at this diagram and kind of discuss how you would read this type of data. First of all, the size of the disk represents how many known species of that group there are. The distance between disks represents how closely related they are. The color also indicates some relative that they're closely related, basically. But you can see the bacteria are over here on the left, all the archaea are here on the right. There's this big blue dot in the middle here called the phylum Cyanobacteria. If you think of cyano as in blue, those are the photosynthetic bacteria. They're actually a small group. I shouldn't say small, it's a large group, but they're photosynthetic bacteria. But you can see that sort of just the layout where they sit tells you how closely or not closely related they are. What we're going to take away from this and from medicine is that, for example, if you have a patient that comes up with a bacterial infection that they don't know how to treat, or perhaps has shown to be resistant to the antibiotics that they've used, rather than picking an antibiotic just out of the blue or trying to test every known antibiotic, which is very expensive, very time consuming, they can look at the relatedness of these organisms. And so, for example, if you have, let's say you have an infection that includes, let's say you have a patient that tests positive for something in the spirochete family, for example, syphilis. So you have a resistant syphilis case. They wouldn't jump down here to use the antibiotic that's used for diphtheria, which belongs to this Corynebacterium group. They would probably jump on the antibiotics that is best well known to treat chlamydia, because these two are much more closely related than these two. And so this is basically how this is being used in medicine now. Now, doesn't mean the doctors are standing around with this picture deciding which antibiotics, but when the guidance for antibiotics comes out, this is the kind of information that they're using now when they make those recommendations for antibiotic treatments, because there are hundreds of different antibiotics. It's almost impossible to test all of them every time you are trying to find the best treatment. So we will talk about other ways that they decide on antibiotics. We'll do the Kirby-Bauer test, which is basically the culture and sensitivity test for antibiotics. But really the take-home message from this chapter is just really make sure you understand these structures and how they operate, what they do for the cell, because they really are the target for a lot of the things we're going to talk about throughout the semester. All right, that's it.